acetyl-CoA, that means that you need 8 additional more ATP. And so if you're going to take into account, then the final cost would be 8 acetyl-CoA, 15 ATP, and 14 NADPHs. If they make it clear where the acetyl-CoAs come from, then if they're coming from the mitochondria, you need to take that into account. If you're not sure and it's vague, I recommend that you go with this answer and then just describe why you got them. So my best advice is if they ever ask you a question of accounting, to always write down why you even wrote that factor in. That way, if let's say we don't care about transport and you accidentally put this in, then we can at least realize that, oh, they knew that this was the cost, they just added an extra cost. And so we wouldn't necessarily go too hard on you on getting that wrong. So I highly advise that if you're gonna make an analysis like this, to write down where that math came from. Say that, oh, the extra 8 ATP came from the transport. That way, if it's wrong, we can at least say, oh, well, we already took that into account, so I'm not gonna ding you for that. For the people who are in the real-time recitation, do you guys have any questions? Okay, also feel free to type it on the chat if you have any questions, if you don't want to interrupt or anything like that. So feel free to type it on the chat and I will try to answer that as well. Okay, so let's move on to the next part then, nitrogen. So nitrogen metabolism involves two things, proteins and nucleotides. I will try to cover the protein one as much as I can. So the nucleotide one is relatively kind of the same instance, a lot of pathways, but I will hopefully focus on what you need to know maybe in the last couple of minutes if we can't cover them. So for nitrogen in our, I guess, bodies and everything, the source of that comes from the air, obviously. And so what is important for us is that we got nitrogen from the plants and animals we eat. But those plants get it from bacteria. And so nitrogen fixation of bacteria convert N2 to NH3. Then NH3 gets converted to NO2 and NO3. And so, and then from these two, plants can make amino acid. And so a lot of times we eat, a lot of our nitrogen comes from the food we eat. We can't really fix back nitrogen ourselves. And so we can't really call ourselves autotrophic in that sense. But just something to note so that you're not just like blindly lost of where the nitrogen comes from. This is not going to be a question in the exam. Just going to give you a background. Also, ammonia is toxic. And so we don't necessarily just engulf ammonia in our food because we would be dead. And so we have to gently take it in the form of amino acids. Some of them we can make, some of them we can't. But in general, we don't chug ammonia. And so there's two known entry pathways for nitrogen. Now, I know I did say that we don't take in ammonia, but there's some ammonia made in the cell, but also a lot of plants, when they get ammonia, they use this pathway. And so these are the two main entryways for nitrogen to enter. So in this reaction, what we're showing here is alpha ketoglutarate combines with ammonia. So essentially, if you have taken 512, this is a condensation between an NH3 and a COO minus, or a CO carbonyl. So hopefully you can remember that when you combine these two, you first form an amine. And then if you reduce this amine, you can get an amine. And this particular amine is special because it has a carboxylic acid next to it. And so this is therefore an amino acid. And so in this reaction to form glutamate, we combine alpha ketoglutarate with the ammonia to form glutamate. And this should be you should start creating a connection between alpha ketoglutarate, AKG, and glutamate. Essentially, I like to think of it as this. Glutamate is the amino acid version. Alpha ketoglutarate is the alpha keto acid version. And so the idea is that the two of them are pretty much the same, except glutamate has the nitrogen, whereas alpha ketoglutarate has the alpha keto acid version. Other examples of these include pyruvate and alanine. So essentially they're the same, except alanine has the nitrogen, whereas pyruvate is the alpha keto acid. And so you want to start, I guess the last one is oxaloacetate and aspartate. So all of these are like twins, if I like to call it that way. So you want to start seeing them like together because the only difference between them is the nitrogen. And then the next entry point is through making glutamine. So in this case, we can actually just use the glutamate we just made and incorporate a nitrogen at the C-terminus by first phosphorylating it. So we take a carboxylic acid and we phosphorylate it. And then after we phosphorylate it, we can have nitrogen attack the carbonyl and essentially kick out this phosphate group so that it becomes an amide group. And now we've incorporated two nitrogens. And so I put in here the cost on the side, just so you know. I don't know if there's going to be a question about cost energy in these pathways, but just in case. Essentially, the NADPH comes in to reduce it. whereas the ATP, because the carboxylic acid and NH3 condensation reaction is unfavorable. You can kind of think about it also in 512. In 512, if you have those, that doesn't necessarily condense. And so we need ATP to make that favorable. And then after we make glutamate and glutamine, these two can now transfer nitrogen to the rest of the cell. And so if some molecule needs nitrogen, then the glutamate and the glutamine can give the nitrogen that they just picked up. But the only way for the glutamate, the nitrogen to enter the, I guess, the biochemistry of the cell is through these two pathways. So keep in mind that these two are very important. Okay, so let's talk about those then. So I said after we make glutamate and glutamine, they will now transfer nitrogen. How does one transfer a nitrogen? So that process occurs via this mechanism called transamination. And so essentially, when you transaminate, you take out a nitrogen NH3 group in one molecule and then you transfer it to another. So the cofactor for that is PLP. So most useful for you to do is if you're ever transferring, so for example, we said earlier that oxaloacetate, when nitrogenated, is aspartate. When alpha-ketoglutarate is nitrogenated, then that is glutamate. And so one potential reaction that we might draw is if, if glutamate and oxaloacetate, if we have these two, and we take the nitrogen from glutamate and give it to oxaloacetate so that this one becomes aspartate. And then this one becomes AKG. In essence, we've transferred nitrogen from glutamate to oxaloacetate. And so if you ever do this kind of reaction, you have to write down PLP because that is the cofactor for that. So PLP looks like this. And it is this carbonyl group right here that can hold on to the nitrogen. And so here, what I'm showing you right here is if this carbonyl binds to the nitrogen. So in this case, if the donor is lysine, basically what will happen is the lysine will react with the carbonyl to form this imine first. And then after water goes in afterwards, the PLP can basically leave with an NH3 group. Professor Bender hadn't covered it much more in depth with arrow pushing in the lecture, so you guys can kind of just look at that for arrow pushing. But essentially what's happening here is that the carbonyl will react with the NH2, and then when it kind of breaks off again, it doesn't just leave as it was originally, it instead brings the NH2 with it. And so that is why this is known to be a carrier. So hopefully you appreciate why we have a carbonyl here. These other groups are not so important in that process, but still important to remember that this is the cofactor that you need. If it's empty, sometimes we call it PMP. If it's carrying something, we often call it PLP. But I think PLP is probably the most important way for you to write it down. So I don't think it's going to show up here because it's in a PowerPoint slide, and I'm playing it as a PDF right now. But you can refer to, I'll upload a version of this where the actual arrow pushing is present, but for the sake of time, we don't have time to discuss it. But kind of what I discussed in the last section is just that if you want to transaminate, the first thing you want to do is charge the PLP with a nitrogen. And then the second thing you want to do is bring that nitrogen that you just picked up into an alpha keto acid. And so for the sake of time, I think that is a mechanism that is much more familiar for you because of 512. And so if you look into the lecture or the textbook, I think you guys can understand that yourself. But essentially, the way I think you should remember it by heart is that there's two phases to it, the charging and then the transferring of that nitrogen into an alpha keto acid. So we're just going to skip on that. So that is essentially how we transfer nitrogen. We use a carrier called PLP, and after we've gotten nitrogen from glutamate and glutamine, we can now transfer that nitrogen anywhere to another molecule and make maybe amino acids out of that. And so that will often show up as like these arrows like this, where something that has nitrogen over here, something that doesn't have nitrogen over here, will now get the nitrogen over here. Now this one will lose the nitrogen. And so if you're ever seeing this kind of reaction where nitrogen is transferred, hopefully you remember to add PLP. Any questions from anyone on real time? Okay, so if not, so this is transfer of nitrogen. Now we're gonna move on to another reaction called the transfer of carbons. So far, what have we learned about adding carbons? We can add carbons by carboxylating a molecule. We can add carbons through transketolase and transalylase. Now we're gonna learn another way to add carbons. And usually this is transfer of one carbon at a time. The difference between this and carboxylation is that carboxylation, the carbon that was added typically gets removed immediately, whereas the carbons that we're going to talk about soon, they often stay. And so these, if PLP is the carrier for nitrogen, the carrier for carbons instead is what's called folates. And so folates, often called tetrahydrofolate or THF. By the way, if you were taking 512 concurrently, this is not the same as the THF that is in 512, tetrahydrofuran. So make sure you differentiate the two. We're not doing a chemical reaction here. So tetrahydrofolate is a carrier of carbons. And then these one carbon groups can be given to molecules if they need a carbon. And so what's so special about this carrier is that you can vary the carbon you're adding. In carboxylation, you're always adding a carbon dioxide. So you're always going to get a COO minus. But if you use a folate, you can add a variety of carbons instead. So let's look at what that looks like. So let's start with tetrahydrofolate as an empty molecule over here. So two important nitrogens is the N5 and N10. So they often are the ones that can hold the carbons. So watch them specifically as we move along. By the way, is this pathway super important for you to remember? No, but what's important for you to realize is how the variety of carbons it can hold. So suppose a serine comes in and donates a carbon. So hopefully you remember serine, if this is my alpha carbon, has a structure that looks like this as an R group. So if serine goes in and donates this carbon right here, it can become glycine. And so essentially it went into it reacted with THF to donate a carbon. And so if it donates this inner carbon right here, then now our THF will have a CH2 bounded next to it. And so this is an example of the reaction where in this case, the donor is serine. Serine donated a methylene group, a CH2 group right here to THF so that now THF is charged with that CH2. So that's an example of one way to get the carbon. Now, this is great. Now you're holding a CH2 group. If something needs a CH2 group, you can give it, THF can give it to somebody. But also, what if you don't want CH2? What if you want a methyl group? And so to make that even cooler, what we can do is reduce this group using NADPH. And if you reduce that CH2 group, it becomes a CH3. Now you have a THF molecule with CH3. Now you can donate a CH3 group to someone. And so this is the start of the pattern where THF, it doesn't matter where it gets the carbon from. It can kind of play around and get reduced, get oxidized, and in the process convert the molecules it holds. That way we can have a variety of carbons we can add. Additional ways is if you oxidize it instead, then you'll get this methvinyl group, which if you add a nitrogen can form this CN double bond. If you dehydrate it, it can become a carbonyl. And additionally, if you don't want to get it from serine, you can also get the carbon from formate. It's not important for you to remember each specific one or the pathway or how it interconverts. It might be important for you to, if you might get asked, how do you go from this to this, to realize that, oh, I'm getting reduced, so maybe I'm going to use NADPH. So it might be important for you to remember that, but this pathway you don't need to the top of your head. It might be important for you to interpret the names. So if I say N5 methyl THF, that tells you that there's a methyl group at the N5 carbon. So hopefully the naming makes sense. For certain groups like methylene, where you need two partners, then it often is written as N5, N10. So when we talk about these molecules, they're often shorthanded. And so for the exam, you can just write them as N5, N-phyl, CH2, THF. You can also just write it like that, as short as you can, so that you don't have to worry about the name. But overall, the whole point of this is that they carry carbons and that they can be interconverted. Any questions? Okay, so here's an example reaction like that. So here we have cysteine, homocysteine specifically. Homocysteine is amino acid, but it's not a proteinogenic amino acid because we don't see homocysteine. We see cysteine, which only has one carbon, but this one has two. So this is homocysteine. This is the precursor to methionine. And the only difference between the two of them is a methyl group. So if you're thinking, how do I add a methyl group to a homocysteine? Well, the only one that can add a methyl group is N5-methyl THF. And so in this case, N5-methyl THF will just donate this group to the SH and essentially make methionine. The exact arrow pushing is not important, but being able to realize when N5 THF is important is what is critical for this exam. In this scenario right here is kind of like a more detailedly drawing version of what I'm saying. Serine can donate this methylene group to THF and get N5-N10 methylene tetrahydrofolate. And so these are just sample reactions. And we'll see more of these when we talk about amino acid synthesis, definitely. Uh,